Today we are going to study about entry of fatty acids into mitochondria. First we'll study how fatty acids are obtained. So sources of fatty acids are three. The first is the fats that we consume in our diet. The second is the fat that is stored in our body as lipid droplets. And the third is fat synthesized in one tissue or organ and transported to another where it is utilized for energy purpose. Some species of organisms use all three sources of fatty acids. Others use one or two. Fats are hydrolyzed into fatty acids which reach the cell where they are utilized for energy purpose. For example, myocyte. Complete oxidation of fatty acid to carbon dioxide and water takes place in three stages. In first stage, oxidation of long chain fatty acid to two carbon units, that is acetyl CoA, takes place. And this process is called as beta oxidation. In the second stage, oxidation of acetyl CoA to carbon dioxide takes place in TCA cycle. And the third is transport of electrons from reduced electron carriers to mitochondrial respiratory chain where they are ultimately oxidized to yield energy. So these are the three stages of fatty acid oxidation. First, we'll study entry of fatty acids into mitochondria. Enzymes of fatty acid oxidation in animal cells. They are located in mitochondrial matrix. Fatty acids with chain length of 12 carbon atoms or less enter mitochondria without the help of transporter, but fatty acids having carbon atoms 14 or more than 14, they are transported into mitochondria by help of a transporter, and this utilizes three enzymatic reactions, and these reactions together form the carnitine shuttle. Because they cannot pass directly into cannot pass directly through the mitochondrial membrane and then enter mitochondria. So they are passed into mitochondria by help of carnitine shuttle. First reaction is catalyzed by family of isozyme. Different isozymes are specific for fatty acids having long chain, short chain or medium chain. So, and these enzymes are present in the outer mitochondrial membrane. These enzymes are acyl-CoA synthetases and they catalyze the following reaction. Fatty acid plus coenzyme A plus ATP will give fatty acyl-CoA plus AMP plus PPI. The enzyme is acyl-CoA synthetase. The formation of fatty acyl-CoA is made possible by hydrolysis of two high energy phosphate bonds, and here in this case PPI is further hydrolyzed by inorganic pyrophosphatase into 2-PI, thus making the reaction go to completion. The overall reaction is therefore written as fatty acid plus coenzyme A plus ATP will give fatty acyl-CoA plus AMP plus 2-PI, and delta G0 prime of this reaction is minus 34 kilojoules per mole. Now, in the second reaction, fatty acyl-CoA esters formed at the cytosolic side of outer mitochondrial membrane can be transported into mitochondria, and fatty acids destined for oxidation, beta oxidation, they are transiently linked to hydroxyl group of carnitine, and this forms the second step to form fatty acyl carnitine, and this reaction is catalyzed by carnitine acyl transferase 1, which is present in outer membrane. Either acyl-CoA passes through the outer membrane and then it is converted into carnitine ester in the intermembrane space, or carnitine ester is formed on the cytosolic phase of outer membrane, and acyl carnitine then enters in, through the outer membrane into intermembrane space. It is not clear by present evidences. Anyway, uh, acyl carnitine is formed and is passed through intermembrane space. It crosses the outer membrane through large pores, which is formed by protein porine, which is present in outer membrane. This is how it crosses the outer membrane and reaches intermembrane space. Then fatty acyl carnitine ester then enters matrix by facilitated diffusion through a transporter. This transporter is acyl carnitine carnitine transporter, and it is present in the inner mitochondrial membrane. In the third and final step of carnitine shuttle, the fatty acyl group is transferred from fatty acyl carnitine to coenzyme A by this enzyme carnitine acyl transferase 2. This enzyme is located on the inner face of inner mitochondrial membrane, and this regenerates fatty acyl CoA and free carnitine into the and releases free carnitine into the matrix. Carnitine re-enters intermembrane space via carnitine transporter. This is the diagram showing entry of fatty acids into mitochondria. The first step is catalyzed by acyl-CoA synthetases, which convert fatty acid into fatty acyl-CoA. Here, in the second reaction of carnitine shuttle, fatty acyl-CoA is transferred to carnitine, forming acyl-carnitine by help of enzyme carnitine acyl transferase 1, which is located on the outer mitochondrial membrane. Then acyl-carnitine crosses the outer membrane, reaches intermembrane space. Then from there, by help of facilitated diffusion through transporter, acyl-carnitine, carnitine transporter, it reaches inner surface or inner face of inner mitochondrial membrane, where the third reaction of carnitine shuttle takes place by help of enzyme carnitine acyl transferase 2. This enzyme transfers acyl group from acyl carnitine to coenzyme A, forming fatty acyl CoA and releasing free carnitine. This free carnitine then diffuses back through the same transporter into intermembrane space and is ready for the next process, next cycle.